In this class, we are going to study about the kingdom Protista's categories. So, according to our NCERT syllabus, there are 5 categories of Protista, and those are the Chrysophytes, and then the dinoflagellates, euglenoids, slime molds, and at last the protozoans. So, before studying about these 5 categories, first of all, let's see a bit about Algae. So when we were studying about the kingdom Monera, there we had studied about a category or a type of bacteria which was called as cyanobacteria, and the other name for that was blue-green alga. And in the kingdom Plantae also, we are going to study about the 3 types of alga, that is green alga, that is called as Chlorophyceae, Rhodophyceae, that is red alga, and Phaeophyceae, that is brown alga. So we are studying about alga in kingdom Monera and then also in kingdom Plantae, and here that is kingdom Protista. Here also we are going to study about alga. So before studying about the various categories of the kingdom Protista, let's just clarify this alga, what this literally is. So first of all, what we have to see here is that alga is a heterogeneous group. So alga is not a very homogeneous type of group. This is the most important thing about alga. So heterogeneous means that various different types of organisms or various different types of properties are present in the members of the kingdom or the category, that is alga. So these alga are normally photosynthetic organisms. So normally the organisms which are present in this kingdom alga, those have photosynthetic abilities, but here also a few are, you can say that these are not exactly photosynthetic, or these are rather parasitic. So normally alga are photosynthetic organisms, and these are characterized by a few properties. For example, the members of alga have the chlorophyll A. Obviously there are other photosynthetic pigments also present in the case of alga, but this chlorophyll A should be there, it is a must. Apart from that, alga members show a unique property, and that is called as non-vascular body or non-vascular structure. Apart from that, these have normal aquatic or semi-aquatic or very moist habitat like ponds or lakes or very moist soil or marshy areas. So alga normally lives in these areas. So these are a few properties about alga, and there is also a very important part, and that is called as non-jacketed sex organ. So when we will be studying the kingdom Plantae after this chapter, there we are going to see that the reproductive structures or the cells in the, or the structures in which the reproductive cells, that is the gametes, are going to be formed, there will be a layer of sterile cells. But here that sterile layer will not be present, and that is why it is called as non-jacketed sex organs. But in the case of higher plants, for example angiosperms, so there we will see that there is a layer of sterile cell around the reproductive structure or the sex organ, but here no such sterile layer will be present, and that is called as non-jacketed sex organ. And we have already discussed about the habitat, and in the case of non-jacketed sex organ, each and every cell is functional, that is, it could contribute in reproduction. But in the case of angiosperms, the sterile layer will not take place or will not take place in the case of reproduction. So it has no role with regard to reproduction. So these are the few properties about the alga, and also in angiosperms we know that after zygote there is a stage that is called as embryo. So embryos are not formed in the case of this category, that is alga. So these are the some of the properties of alga. Now first let's see where alga are present. So alga are normally present in 3 categories or 3 kingdoms. So in kingdom Monera, we see a form of alga which is called as blue-green alga. And here in this kingdom, that is, we are studying about Protista, here also alga are present, and these are called as Protistan alga. So we will study about its categories later. But first of all, let's see who are the members of the Protestant alga, and these are the diatoms which come under Chrysophytes and also the euglenoids. Apart from that, there is a category which is called as dinoflagellates, and after that there is also a small category that is called as yellow-green alga. So compare it with it, that is blue-green alga, and here we have yellow-green alga. So don't confuse between these two. So we don't have to study much about this yellow-green alga, but we have to study about these three categories in detail, because in kingdom Protista these three categories come under this category, and that is called as Protestant alga. So all the diatoms, dinoflagellates and euglenoids, these are studied under this category, that is Protestant alga. And when we will be studying the next chapter, that is kingdom Plantae or the plant kingdom, there we are going to see that this alga will come again, and there also three types of alga will be there: Rhodophyceae, Phaeophyceae and Chlorophyceae. So Rhodophyceae means red alga, Phaeophyceae brown alga, and Chlorophyceae, that is green alga. So you can see that this alga is a heterogeneous group. Why? Because it has various types of members, and which are so diverse, so different from each other, that the members of alga are present under different kingdom. And we know very well that the members of a kingdom are pretty much different from another kingdom. So you can imagine now how much the members of alga differ from each other. Now with regard to this chapter, because we are now studying the kingdom Protista, so here with regard to this we have to study about a certain property. So we normally imagine that photosynthesis is done mostly by the green plants, and it is true also, because photosynthesis is really done by the green plants. But one thing that we do not commonly realize is that 80%, or remember always, 80% of the photosynthesis is done by the Protestant alga. Now that is here, Protestant alga perform about 80% of the total photosynthesis on earth. And also you will study about a category that is called as phytoplanktons. So these phytoplanktons are basically very minute or microscopic, and these are single celled structure normally, and these float passively on water surface. Now this word that is phyto, is used in the case of plants or for something that is similar to a plant. So phyto and plankton means that it has certain plant-like properties. For example, photosynthetic ability. So these are normally found on the surface of water, and these float passively. And out of the total phytoplankton population of the world, the Protestant alga, and who are the Protestant alga? These four categories, that is diatoms, dinoflagellates, euglenoids and yellow-green alga. These comprise about 95% of the population of the phytoplanktons, and these categories perform the 80% photosynthesis on earth. So you can imagine how much these are important for each and every organism, because ultimately this photosynthesis is not like bacterial photosynthesis where oxygen is not released. This photosynthesis is oxygenic. Oxygenic means it gives oxygen as byproduct. So now you can imagine how important this category, that is the Protestant alga, is. And in this chapter we have to study about three categories of Protestant alga, and that is Chrysophytes, dinoflagellates and then the euglenoids. So these are the three categories of the Protestant alga that we have to study according to our NEET syllabus. But also there is a small category that is called as yellow-green alga. So it is also a Protestant alga, but we don't have to study much about those categories because that is not much included in our syllabus. So we are going to limit our discussion to the Chrysophytes, dinoflagellates and euglenoids in the category of Protestant alga. But after these three categories, we also have to study about two more categories, and that is the slime molds and also the last one that is very similar to animals, and that is called as protozoans or protozoa. So before moving ahead, we have to study about a bit more about planktons. So there are two types of planktons. One is phytoplankton and the other one is the zooplankton. So this phytoplankton does have plant-like properties. Therefore, it is photosynthesis or it produces food. And the other one that is called as zooplankton. So we know that that zoo word is used for animal-like properties or for animals. So this phytoplankton is the producer and the zooplankton is the primary consumer, and ultimately zooplanktons are eaten by certain fishes or the crustaceans. So ultimately this phytoplankton is the major producer in the case of oceans. So after this fact, we have to study about the first category according to our NCERT, and that is called as the category which is called as Chrysophyta. So the members are normally called as Chrysophytes, and the division, that is, we know after kingdom the category that comes is the phylum or the division. So this is the division, that is Chrysophyta, and the members are called as Chrysophytes according to our NCERT. Now the first thing to remember is that it includes two organisms and two categories actually, and that is the diatoms and the other one which is called as golden alga or desmids. So these two categories are normally found only in water, but in water also we know that there are two types of environment. For example, sea or ocean like marine environment, and lakes, ponds or rivers like freshwater environment. So these two categories are found normally in each type of environment. So diatoms mostly occur in the case of marine environment or marine habitat, but it is not that any diatom does not occur in freshwater. So it is being discussed about their majority group. So the majority of diatoms live in the marine environment, and the majority of the desmids, which are also called as golden alga, so these are normally found in fresh water. So that is what is being written here. So so they are found in fresh water, mainly desmids, that is in fresh water mainly golden alga are present, and in marine that is mainly diatoms are present. After this we have to study about their various shapes. So in bacteria we studied about something called as mycoplasma. So mycoplasma were pleomorphic. So pleomorphic means there are a lots of different types of shapes in that category, and here also we have a variety of shapes here. For example, circular, rectangular, triangular, and also elongated, spherical. So various different types of shapes are possible over here. After this we have to study about a few properties that we already studied here, that is the phytoplankton. So these are microscopic because these are normally consisting of a single cell. So a single cell is obviously very small normally, and that is why it is microscopic, and we know that it is photosynthetic because we already studied here. So this category, because it is coming under protestant alga, we have studied that it is photosynthetic. And obviously we saw that these phytoplanktons are normally present on the surface of water, and it is saying here that these are the chief producers of oceans. And why these are chief producers of oceans, not the plants? Because in the case of oceans which are so deep, how can plants even survive there? So obviously plants are not present normally in oceans. So these are the chief producers, not the green plants. But we know that on land, that is terrestrial habitat, plants are also one of the most important producers. But these are the chief producers of oceans. And after the phytoplanktons, the category which is the consumer, that is called as zooplankton. So zooplankton eats phytoplankton, then fishes and crustaceans eat the zooplankton, then fishes are eaten by larger fishes, and there also the food chain continues like that. So after that we have to study about that these don't have flagella. And now we have to remember here that if an organism does not have flagella, so it will not be able to move by itself. So in our previous classes we had studied about something called as mucilage propulsion. So when we were studying about the locomotion in the kingdom Protista, we studied various types of locomotion. But here we don't have flagella, but there are certain other methods, and that is called as mucilage propulsion. So what happens? It is very simple. Just like a rocket, from a rocket gases come out from one end and the rocket is propelled. Same way, mucilage comes out from one end. So this mucilage is ultimately a sort of carbohydrate, and when this mucilage comes out from one end, the Protista or this in category which is called as Chrysophytes, the members are propelled through or in one direction, and that is called as mucilage propulsion. So they don't have a flagella and hence they float passively. Passively means not by their own will. So that is what is meant here, passively with water current. But this is not the case with all of the phytoplankton. Some have gliding motion by mucilage secretion. And what was the type of mucilage secretion? We just wrote here, mucilage propulsion. After that we have to study about some of the reserve food. So we know that we are humans, animals. So in humans, glycogen is the stored food. In green plants or the angiosperm or gymnosperm, starch is the reserved food. Likewise, in alga, we are going to study about that there are certain types of foods which are stored. So ultimately we have leucocin. So this leucocin is also a polysaccharide of beta glucans. So we are going to study about glucans in the chapter that is the biomolecules. So we know about a few polysaccharides, for example cellulose or the glycogen or the starch. So likewise leucocin is also a polysaccharide, so we can say that it is also a carbohydrate. Likewise we have chrysolaminarin. So when we will study about the kingdom Plantae, there we will study about this laminarin, and it is also a stored form of carbohydrate. And here we are studying the category which is called as Chrysophytes. That is why it is written chrysolaminarin. So we will study about this chemical again in the kingdom Plantae, and under what? Under alga. So here laminarin plus chryso, that is chrysolaminarin. Why? Because we are studying the category which is called as Chrysophyta or the Chrysophytes. After this we have to study about that these have also some fat drops. So we know that fats are pretty light. So the fats are helping the plant or this phytoplanktons to float on the surface of water. So we already have studied that they float passively with water current. So we know they must be very light. That is why they are floating on water. And why they are so light? So we know now the answer. So these are floating because these have fat drops. So these fat drops are helping this phytoplankton to float passively on the surface of water. Apart from that, we also have to remember one very important fact, that out of the phytoplanktons, that is 95% of the phytoplanktons, almost 50% or even more than 50% of the phytoplanktons are constituted by the diatoms. So it is a very important point. So 95% of the phytoplanktons are consisted of diatoms, dinoflagellates, euglenoids and yellow-green alga. And out of these, that is this 95%, more than 50% of the phytoplanktons are constituted by these diatoms. So now we have studied about a few points about the chrysophytes. And now we have to study about a few more points regarding this category, that is division Chrysophyta. Now we are going to study about a few more points about the category Chrysophytes. So we know that the Chrysophytes belong to the category that is the Protistan alga. So it is a photosynthetic category. So for photosynthesis there must be the presence of a pigment. So the pigment which is present here is the chlorophyll A, chlorophyll C, and apart from that there are some minor pigments which are called as carotenoids or the fucoxanthins. So this fucoxanthin will again come in the category that is brown alga, and we will study about the brown alga in the kingdom Plantae, and there we have to study about alga again. So these are the photosynthetic pigments, and most important is chlorophyll A. So in short, chlorophyll is written as CHL. So this chlorophyll A is the most important pigment, and thereafter chlorophyll C, and then carotenoids and the fucoxanthins. So these are the photosynthetic pigment present in the case of this chrysophytes. So now we come to the main part, and that is the cell wall or their structure or composition in the case of diatoms. So it is a very interesting thing, and here first of all we are going to see that their cell wall is made up of two parts, which are called as epitheca or the hypotheca, or the epivalve or the hypovalve. So normally we know that in the case of plants, the cell wall is composed of mainly cellulose, but here cell wall is composed of cellulose, some pectic compounds, and also a very resistant substance, very hard substance, and that is silica. So this silica particles are deposited in the cellulose or the pectin layer or the pectic compound layer present in the cell wall of chrysophytes. So the cell walls are made up of two halves, and we are going to see that these are the two halves. So the outer and the bigger and the larger is called the epitheca or epivalve. So these are the two halves, the larger and the older. The larger one is also the older one. We will see later why. So one part is called as, which is the larger and the older, that is the epivalve, and the smaller one, that is the smaller as well as the younger, and that is called as epitheca or epivalve. And these fit together like a soap box. So you must have had various soap boxes in your homes, and we know that the soap boxes, two parts of the soap boxes fit together like this. So in this two part, that is the two parts of the soap box, here silica are embedded or deposited. So the cell wall is made up of cellulose. We now have seen pectic compounds in which silica particles are embedded, and these are two parts. There are two parts of this cell wall, and the cell wall which is having silica deposits, that is called as silicated, and that is named as frustule, or also that is called as shell. So that is called as shell or frustule. So we can see here that there are two parts of this frustule or shell. Frustule has two parts. That frustule is another name for the cell wall of this protista or the chrysophytes. Now it has larger, older, that is this part. This is the larger and the older part. It is called as epitheca or epivalve. Now we have a smaller, younger, that is it is synthesized later. And this smaller, younger one is called as hypotheca or the hypovalve. You can see here this is a smaller one and this is the larger one. Now we know that silica is a very hard and very resistant chemical. It does not get destroyed very easily. It can survive for millions of years. So if silica is deposited in the cell wall of chrysophytes, we know that even after the death of chrysophytes, the silica will remain, and because of this property of chrysophytes, huge deposits of silica have been formed, because we know that after that they will sink to the bottom of the sea and they have silica in their cell walls. So in millions of years, these deposits have become very huge, and those deposits or those rocks which have been formed by the dead diatoms, that is called as diatomite, or it is also called as kieselguhr, or it is also called as diatomaceous earth. So how is that being formed? So it has been formed because of deposition of the dead diatoms over millions of years. So because of the presence of silica, the cell wall is very hard and does not get destroyed after their death. It's very simple, silica does not get destroyed easily. Now therefore at the bottom of sea very huge rocks of dead diatoms have been formed over millions of years, because these organisms are very old, much more older as compared to humans or the other animals. So over the millions of years, these have been deposited over the bottom of the sea, which is also called as the seabed or the ocean bed, and there it is called as diatomaceous earth or kieselguhr or the diatomite. So these are the three names. Now these structures or these rocks or their fragments have very fine pores in them. So because of the presence of those very fine pores, this could be used for filtration of oils or syrups and other things like beer. So it is used normally for filtration. Apart from the filtration, it has various other uses, because this silica or the diatomaceous earth is an insulator. It is not a thermal conductor. Therefore it can be used as an insulator in steam boilers. Apart from that, there are lots of other uses of insulating material, and there this diatomaceous earth could be used. Apart from that, it is also used in stone polishing. Apart from that, it is also used for soundproofing. So there are certain recording studios which need to be soundproof, so or big halls which need to be soundproof. So there, in the case of soundproofing, this diatomaceous earth could be used, and also as an insulator or stone polishing. So we know how important economically or commercially this diatomaceous earth or diatomite is. Now this is about the commercial use of the diatomite or the diatomaceous earth. Now we come to the reproduction. So reproduction is mostly by the asexual reproduction, and in asexual reproduction the most common method is the binary fission. So we will study later, this is a normal sequence or this is the normal diagram of binary fission, and we will study this later. Each daughter receives one valve. Now we see that there are two valves, epivalve and hypovalve. So when binary fission will occur, each daughter will receive one valve, that is one epivalve, one daughter will receive, and the other daughter will receive one hypovalve. So now we will study that with the help of this diagram. So suppose this is the parent, and in the parent we know that the bigger one is the older one. From here we know that the larger one is the older one. So there are two ones. So we are going to denote the older with O and the newer with N. So this red one is here is the newer one, and this black one is the older one. Now according to here, each daughter receives one valve. So this is one daughter, this is one daughter. Now this daughter will receive one older one. So we can say that this larger one is the older one, and it will synthesize a new one. So this black one is the older one, and red one is the new which has been synthesized, because we know that the cell wall will consist of two parts. So it has got one part from the parent, and that is the black one, which is the older one, and now it has synthesized a new part, that is the hypotheca or the hypovalve. So this red one here is the new one. But let's take the example of this daughter. These both are daughters of this parent. So it has received this red one as the older one, because this one has been obtained from the parent. So this red one is the older one, and this black one has been synthesized newly. So this black one is new. that is here, this black one is new, this red one is old. It is reverse of the above. You can see here the black one or the larger one was the older. Here red one is the newer one. Here a smaller hypovalve has been synthesized, and here a smaller hypovalve has been synthesized. Now this each valve that has been obtained from the parent. So this black one has been obtained from the parent, this red one has been obtained from the parent. These both function as epivalve, because you can see the new valve which has been synthesized is the smaller one. So the smaller one is called as hypovalve. So this is what is occurring over here. Now, and synthesis of hypovalve takes place. So here it received the black older one and it synthesized a new one. Here it received the red one and synthesized a new smaller black one. So we can see here that this results in size reduction over successive generations. So you can observe it here. So one is larger, the other is smaller, that is obvious. So this is the larger one, it has synthesized a smaller one. But this is a smaller in size as compared to the parent. You can see this why. Because the smaller or the hypovalve of the parent is now the epivalve of the daughter, and obviously the new hypovalve will be the smaller of this. So obviously there has been size reduction, but here there is no size reduction. In this daughter, no size reduction. Why? Because the epivalve of the parent is also the epivalve of the daughter, but here the hypovalve of the parent is the epivalve of the daughter. Now this daughter will also reproduce after some time. Here you can see this older one has been given to this one. So this black one is the older valve and it has synthesized a new one, that is this red one. So this red one is the new one. Now this red one is now the epivalve of this. So this is the older one and this is the newer one. So you can see no size reduction from here to here, but from here to here there has been size reduction. Now same goes for this. This red one has been transferred to this one. So this red one is the older here and this black one is the newer here. So there has been no size reduction. But now here what is happening is that this new one is now its epivalve and it has synthesized a new, that is called as the hypovalve. So this is the older one and this is the newer one. So you can see here there has been size reduction, but here no size reduction from the parent. So we can see that in one daughter there will be size reduction and in the next daughter there will be no size reduction. So from here to here there has been consistent size reduction. From here to here size reduction, from here to here then also size reduction. So over successive generations we are seeing that even it will, after a few generations it will become even more smaller. So obviously at a certain time this will almost disappear or it will rather be not able to live. So this is what is occurring here. So it is a bit more complex to understand, but it is very simple once you understand it. So there could be size reduction in one daughter, there could be no size reduction in the other daughter. That is why, so which daughter, the daughter which will receive the epitheca or the epivalve, it will not have size reduction. The daughter which will have the or which will receive the hypotheca or hypovalve, that will have size reduction. We can see this daughter has received the epitheca, black one, that is why no size reduction. This daughter has received the hypotheca of the parent, that is why size reduction. Same goes for here. This daughter from this parent has received the epitheca, so no size reduction. This daughter has received the hypotheca of the parent, therefore it has undergone size reduction. Now here also this daughter has received the hypotheca or the epitheca of the parent, so no size reduction from here to here. But in this case there has been size reduction from here to here, because this daughter has received the hypotheca or the hypovalve of the parent. Therefore, this daughter will be much smaller as compared to parents. This daughter will be smaller as compared to his parents. So that is why over successive generations, due to binary fissions and due to the difference in the epitheca and the hypotheca, the size will go on reducing. So there has to be a mechanism to increase the size to the normal, and that mechanism is the sexual reproduction. So we know binary fission is an asexual reproduction, and zygote formation takes place from sexual reproduction or fertilization. So when we are talking about zygote, we know we are talking about sexual reproduction normally. So this zygote is normally a product of sexual reproduction, and these zygotes develop into something called as oxospores. So these are rejuvenescent spores. So rejuvenescent here means that it will increase the size to the normal. So binary fission results in decrease in size over the generations, but sexual reproduction through the oxospores or the zygotes, it increases the size to the normal. So after a certain number of generation, the size has been reduced to very small, and then this zygote formation, and therefore the oxospores will be formed. And after this oxospore, the size of the offspring will again increase to normal. That is something like this. So we can say that this is a rejuvenescent spore because it helps in increasing the size of diatoms to normal, because after certain generations it will become much smaller, and due to the oxospore formation, because of the sexual reproduction, the size will increase again to the normal or the initial size of the protist in here, the chrysophytes or the diatom. So this is what is oxospore, and here we are talking about sexual reproduction, and here we are talking about asexual reproduction. So binary fission reduces the size, and this oxospore formation increases the size. So these two processes are almost opposite to each other. So now we have completed the reproduction in chrysophytes and also the diatom. So this is what we have to study about the diatoms according to our syllabus. So now we have completed the first category of protista. So these were the chrysophytes, and after this we have to study about dinoflagellates, euglenoids, slime molds, and also the protozoans.